There are two ways the cell can die: necrosis and apoptosis. Necrosis occurs when a cell is damaged by the external force, such as poison. Apoptosis, on the other hand, is a form of programmed cell death that occurs in multicellular organisms. It is a regulated form of cell death and has a role in biological processes including embryogenesis, aging, and many diseases. Apoptosis can be triggered in a cell through two main pathways, intrinsic and extrinsic. The extrinsic pathway is initiated through the stimulation of the transmembrane death receptors located on the cell membrane. In contrast, the intrinsic pathway is initiated through the release of signal factors by mitochondria within the cell. Apoptosis is genetically highly regulated and it clears out damaged or cancer cells. Caspase, which is also known as cysteine-dependent aspartate-directed proteases, are a family of genes important for maintaining homeostasis through regulating cell death and inflammation. Caspases are synthesized in the cell as inactive precursors and are activated only during apoptosis. In addition to apoptosis, caspase 8 is also required for the inhibition of another form of programmed cell death called necrotosis. The initiator of apoptosis proteins, which is also known as IAPS, are a family of functionally and structurally related proteins that serve as endogenous inhibitors of programmed cell death. A common feature of all IAPs is the presence of BIR, also known as baculovirus IAP repeat. The best characterized IAP is XIAP, which binds to caspase 9. The initiator caspase is initially made in an inactive monomeric form, sometimes called a pro-caspase. Apoptotic signals trigger the assembly of adaptive proteins that carry multiple binding sites for the caspase amino terminal domain. The initial caspase are activated after dimerizing from binding to adapter proteins. When the active caspase is cleaved at a site in the protease domain by an initiator caspase, the executioner caspase dimer goes through conformational change to activate it. Trimeric FAST ligands on the surface of killer lymphocytes will bind to the FAST receptors on the surface of a apoptotic cell. This leads to receptors clustering, which activates death domains on the receptor tails, which interact with similar domains on the adapter protein FAST-associated death domain, or FADD. The adapter protein then recruits an initiator caspase, caspase 8, which forms a death-inducing signaling complex, or DISCs. Within the DISCs, two initiator caspases dimerize and cleave itself between the protease and the death effector domain. This then releases active caspase dimer into the cytosol, where it activates executioner caspase by cleaving them. This lengthy process shows how tightly regulated apoptosis is. In this research conducted by Dr. Silk and Dr. Mir, it was concluded that IAPs contribute to cell survival at multiple levels, and it is a very important protein for the proper functioning of apoptosis. This conclusion is based off their research that proved the alterations in IAPs were found in many types of cancer and are associated with disease progression and poor prognosis. However, a deeper understanding of the IAP mechanism needs to be studied to further expand our knowledge in this topic. Also, another possible research topic is in what ways inhibition of IAP could be beneficial. Perhaps inhibiting the function of IAP and letting the apoptosis to be fully activated could be beneficial in some cases as well. Thank you!